I'm Juliana.
I'm Annalise, and this is our muscular system tutorial.
The three main types of muscle cells are skeletal, cardiac, and smooth. Cardiac cells are found in the heart and are responsible for pumping blood. They are under involuntary control. Skeletal cells are attached to bones and tendons and are responsible for voluntary movement. Smooth muscle cells are found in the walls of hollow organs like the liver, the pancreas, and the intestines, and blood vessels, and are responsible for controlling functions like digestion and blood flow. They are under involuntary control.
Moving on to the three muscle types in rats.
Here is an example of rat cardiac muscle from our rat dissection through the dissection scope. Here is skeletal muscle that we got from the quad muscle of the rat. Here is smooth muscle that we took from the intestine.
Now moving on to the three muscle types under the microscope.
Right now we're looking at a picture of smooth muscle under a microscope. It appears homogeneous and it has a large amount of actin and myosin, which we'll get to later. The cardiac muscle appears striated and it has intercalated discs which connect the cells. They also have numerous amount of mitochondria that supports its high energy demands. Skeletal muscle is striated and multinucleated. It's multinucleated to enhance oxygen uptake and to make tissue repair more efficient. Through this experiment, we discovered reflex blinking. It's a type of involuntary blinking when something gets too close to your eye. Your corneal reflex kicks in whenever you sense danger from an elicited stimuli. We're going to be talking about how the nervous system controls muscle movement using this image. Movement starts in the brain, where a signal or a nerve impulse is generated in response to decision or a reflex. The nerve impulse travels down the spinal cord and through motor neurons towards the muscle. This is shown as the signal pathway from the brain to the arm. At the neuromuscular junction, or the area where the motor nerves connect and communicate with the muscle, motor neurons release neurotransmitters like acetylcholine, which trigger the muscle fibers to contract. The muscle fibers respond to the signal by shortening or contracting. This allows the arm to move and perform the desired actions, such as catching or throwing the ball.
What allows for these muscle contractions to occur?
Here are two muscle fibers. The first, with ATP and salt, is shorter because ATP powers the sodium-potassium pump, which maintains the ion balance needed for contraction. Salt provides the sodium and potassium ions required for this process. The second, with salt but no ATP, is longer because the pump can't function without ATP, disrupting the ion balance and preventing proper contraction. This shows that both ATP and salt are essential for muscle contraction to occur.
Which solution is best for muscle contractions? The best solution contains salt and ATP, and you'll see why after we describe all the muscular structures. Beginning with the tendon, this connects muscles to bones, and the deep fascia supports and protects muscles and other soft tissue structures. The epimysium is a protective layer sheet that protects from friction. Inside the muscle tissue, there are perimysium, which contain the muscle fibers or fasciculi. And around the perimysium, there are tons of blood vessels that help supply blood in the muscle. Each fascicle is made up of bundles of muscle fibers, and the muscle fibers are contained by the endomysium. Each muscle fiber cell has motor neurons and blood capillaries that help take waste away from your muscles. Each myofibril can be broken down into sections such as the sarcomeres, and this is the basis for sliding filament theory. Now we're starting at muscle contractions at the nerve cell. Excitation-contraction coupling is a mechanism by which muscle fibers translate an electrical stimulus into a mechanical response. This is how muscles contract. As a nerve impulse reaches the ends of an axon terminal, it stimulates the release of acetylcholine from synaptic vesicles. Acetylcholine then diffuses and attaches to the acetylcholine receptors on the sarcolemma of the neuromuscular junction. After acetylcholine attaches to the receptors on the sarcolemma, the muscle fibers become more positively charged, which generates an action potential and travels down the sarcolemma and transfers T-tubes. This action potential releases calcium from the sarcoplasmic reticulum.
Calcium binds to troponin on the actin filaments of muscle fibers. This signals tropomyosin to move out of myosin binding sites on actin. Energized myosin heads with an attached ADP molecule and phosphate group bind to the open myosin binding sites on actin. This forms the actin-myosin crossbridge. After the crossbridge forms, the myosin head rotates towards the center of the sarcomere. As it rotates, the myosin releases its ADP and phosphate. This action slides the thin actin past the thick filament, creating the power stroke. After the power stroke, the actin-myosin crossbridge remains intact until an ADP molecule binds to the myosin head. After ATP attaches to the myosin detaches from the actin.